Hello, everyone, and welcome to today's live broadcast, Development of a Novel Method to Measure Circulating 125 Dihydroxyvitamin D in Human Serum and Plasma. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by DSORIN Incorporated. DSORIN Incorporated offers the broadest, fully automated infectious disease and specialty diagnostics menu, including best-in-class vitamin D. These quality assays are analyzed on the versatile LIAISON XL platform. They provide value through sales, service, and support solutions to hospital and reference laboratories by consolidating their menu in order to streamline processes and increase efficiency. For more information, please go to www.dsorin.com. Before we begin, just a couple of important announcements. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit these questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. All questions submitted during the webinar will be answered via email after the presentation. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Fabrizio Bonelli. Fabrizio Bonelli is Chief Scientific Officer of the DSORIN Group. He has been working in the R&D department of DSORIN for 25 years. I apologize, that's 29 years, and previously at the Italian site and since 2010 at the U.S. site in Minnesota. Prior to joining DSORIN, he was postdoctoral fellowship at the National Cancer Institute of Bethesda, Maryland, conducting research in the field of cellular carcinogenesis and tumor promotion, sepsis, endocrinology, and maternal fetal medicine have resulted in multiple publications and awards. I will now turn it over to Dr. Fabrizio Bonelli. Dr. Bonelli?
Yes, as Christina was saying, today I'm going to talk of a novel method that, to measure the 125 vitamin D in serum and plasma. And I would like to start with some background information about the vitamin D. Vitamin D is produced through a series of events. So basically UV light from the sun will convert the 7-dihydroxycholesterol in pre-vitamin D3. And then the pre-vitamin D3 will be converted to vitamin D3. And then at the liver level, the vitamin D will be converted in 25-hydroxyvitamin D that in the kidney will become the 125-hydroxyvitamin D. And here in this slide, there are the two metabolites, the main metabolites of the vitamin D, so the 25-hydroxyvitamin D, which is the major circulating form. The concentration is at nanogram per ml. From the biological standpoint, is inactive, is not regulated, and the half-life is from 2 to 3 weeks. Whilst the 125 is the active hormone, is a steroid hormone, circulate at much lower concentration, so picogram per ml rather than nanogram per ml, is biological active, is highly regulated, and the half-life is much shorter, just 4 to 6 hours. So what the 125 does is, being the hormone binds to its receptor, is called vitamin D receptor or VDR, and the VDR, when it is bound to the vitamin D, the 125 binds to the DNA to certain region called VDRE, vitamin D responsive element, and in this way regulates target genes. So here I'm just briefly mentioning which is the physiological significance of the 125. So one of the well-established roles is in the bone and mineral metabolism, in concert with another hormone that is the parathyroid hormone, or also called PTH. And basically, the concerted action of the 125 dihydroxyvitamin D with the PTH is to make sure that the plasma concentration in the plasma is regulated. So this is achieved via intestine, meaning that the vitamin D 125 promotes the absorption of the dietary calcium from the gastrointestinal tract. The second action is about the reabsorption of the calcium at the kidney level, thus reducing the calcium in the urine. And the third one is to promote the reabsorption of the calcium, if when needed, from the bone through the action on some cells called osteoblasts. And then the plasma calcium eventually will be used for the mineralization of the bone, as well as will be used at neuromuscular level. In the next slide, here is shown like as the calcium is regulated by this interaction between PTH 125 and calcitonin. So the serum calcium, when is low, it requires to be increased, and this is done by the 125 that is, as I said, stimulate the reabsorption at the bone level, at renal level, at the intestinal level. And the PTH, when the calcium is low, is inducing the production of the 1-alpha hydroxylase that is using the 25-hydroxy reservoir to make the 125. So when the physiological role doesn't work, there is something, some consequences at pathological level. So when you have low 125 vitamin D levels, and what you may have are disorders in 1-alpha hydroxylation, and this may link to chronic renal failure, or the vitamin D dependent rickets type 1, and the hypophosphatemic rickets. Another consequence of the low 125 is the hypoparathyroidism, and the third one is the severe vitamin D deficiency that may recur in many different pathological situations. When you have high 125 vitamin D, you may have another type of rickets, it's called vitamin D rickets type 2, or you may have primary hyperparathyroidism, and also another disease that is associated to the elevated 125 levels are due to extrarenal 1-alpha hydroxylation that is present in patients with sarcoidosis, inflammatory bowel disease, and lymphoproliferative diseases. So let me say that 125 is not commonly used in the lab for assessing this, particularly the low vitamin D status, simply because the assay has been very difficult. So an assay for the vitamin D needs to be challenged by three main issues. First of all is the low circulation level of the 125, as I said at the beginning, picogram per ml rather than nanogram per ml. Similarity to the 25 vitamin D, which is in nanogram per ml. So it's similar and is in lower concentration. And in order to measure the 125, all the methods are using pre-analytical step to make sure to enrich the 125 content over the 25 D content. So what we did in order to overcome this limitation was to develop an assay that was designed based on the following pillars. First of all, we use a recombinant ligand-binding domain called also LBD, which is derived from the VDR, and we have used the LBD as 125 capture molecule. The second pillar is the exploitation of a conformational change of the LBD induced by the 125D binding. The third one is to use a reaction condition in the assay which favor the 125 to the LBD. And the fourth characteristics pillar is the use of the monoclonal antibody which recognize the conformational change. So starting from the LBD, a few years ago, the group of De Luca, the University of Wisconsin, generated a recombinant polypeptide which was derived from the portion of the VDR encompassing the ligand binding domain. And more recently, a team also led by De Luca with Singarapu as a first name in biochemistry, they reported on the paper that the solution of NMR of the LBD show significant spectral differences with and without the ligand. And these differences suggested the idea that different conformation may be used to elicit specific immunological response against each form, thus being able to discriminate between the two forms by immunological methods. So here, in the next slide, I'm just reporting briefly which was the outcome of the study of Singarapu. Here you see on the left-hand side the LBD without the ligand, and basically there is no any significant resolution. Whilst when you add in solution the 125 vitamin D, you start to see some secondary structure, and in particular there is the helix 12 of the LBD that in which the flexibility is reduced, thus allowing to solve structures. So these two graphs figure are showing that there is a difference between the LBD without and with the ligand. So the next characteristic was about the reaction condition. So there have been published in the past study by which the 125 has 200-fold higher affinity to the LBD compared to the 25 at the reaction condition that we have selected in our assay. In the opposite, the 25 vitamin D has from 10 to 100-fold higher affinity for the vitamin D binding protein as compared to the 125 dihydroxyvitamin D. So in total, as a result, we estimate there are 2,000 from 2,000 to 20,000-fold binding differential in terms of affinity between the 125 compared from the 125 to the LBD as compared to the 25. The next step was the development of the specific monoclonal antibody. The antigen-specific immune response was determined by ELISA using plates that were based on using the immunogen of interest, so meaning the complex LBD 125. And then there was the plate, the hybridoma cell line were counter-screened with the free LBD. So the absorption of the LBD 125 of the complex was done on microtiter pair plates that was containing nickel ions to make sure to be able to capture the protein that has a fusion domain with the polyhistidine. And this is to make sure that the complex was properly oriented and available for the binding. And by using this strategy, we were able to isolate clones that were specifically directed towards the complex rather than the non-complex LBD. And in particular, one of these clones was highly reactive. His name was 11B4. And this line has been selected for additional characterization and then afterwards for the use in the final assay configuration. So here, what we did in the previous phase of development was to show that indeed the 11V4 is able to specifically recognize the complex rather than the LBD. We used to do that a biosensor with called DPI, dual polarization interferometry, and what we did with a 2-channel chip, we coated the chip with the monoclonal antibody, and we were flowing in the chip. In the blue part, we were injecting the 125 complex. In the red, we were injecting the LBD only. And as you can see, there is a huge difference between the complex and the LBD only. That's demonstrating that the monoclonal recognizes specifically the complex only and does not recognize the LBD alone. Then the next step was to sequence the monoclonal, and to do that, what we create from the hybridoma of the 11V4 cell line, a cDNA library and we were able to clone the heavy chain as well as the variable chain, and we clone in a vector through the TA cloning method. And we isolate 20 independent bacterial colonies, and then what we did was sequencing them, and upon sequence alignment, we isolated one specific representative VH and VL chains, and indicating the hybridoma population was indeed monoclonal. And here you have the sequence, I know that is a bit hard to see, of the light chain as well as the V chain, with the framework region as well as the hypervariable region that are indicated in red, whilst the framework regions are indicated in green. So once we have the monoclonal and we show that clearly the monoclonal does recognize the complex, we develop the assay method that we call Receptor-Assisted Chemilumiscent assay, or REACH assay. So how this assay works? So the step one is to incubate the sample with the assay buffer. The sample will contain the binding protein with either the 25 vitamin D or the 125 vitamin D. Here depicted in green, the 125, and in yellow, the 25. And as I said at the beginning, the great majority of the vitamin D is the 25 rather than the 125. Then we had the assay buffer, which with the certain pH that is favoring the binding of the 125 to LBD compared to the 25. And we had the ligand binding domain. The recombinant binding domain at this point is free. And then we incubate. So in this phase, what we would expect that the 125 will be binding the LBD, and upon binding, we change the conformation. Once this first incubation has been completed, then we add the paramagnetic particles, which contain the monoclonal antibody against the complex, against the LBD complex with the 125. And we incubate again for a certain time. And during this phase, we would expect the monoclonal will be able to capture the LBD with the 125. After this step, we wash, and then we add the second step in which, after washing, we add the monoclonal antibody that contains conjugated with our signal, which is the aminobutyl isoluminol, that will recognize the LBD, a fragmented LBD, specifically just to make the snapshot, the recognition of the binding which was occurring in the first step of the assay. After the second incubation, we have the wash, the second wash step, and at this point we read the signal that will be developed once the beads will be separated in the instrument. And as you can see, the type of signal that we see with this specific assay is a signal which is growing with the dose of the 125. And we define this methodology, an assay for a small molecule which is non-competitive as compared to other methods for detecting small molecules that are based on the non-competitive format. And by using this methodology, the assay, as you will see in a moment, is much more precise of any other method that is using instead the competitive format. And this assay is quite fast in terms of the first time of the results. So we, in total, all the incubation will require 65 minutes. And then the throughput after the first incubation is about 80-90 samples per hour. So to summarize the assay characteristic, this is the first assay which is being for 125 that is fully automated, does not require an extraction step. It requires also a very limited volume of sample, 75 microliters, and 65 minutes for the time for the first result. So now I'm going to show you some analytical characteristics of the assay. First of all, assay precision. And the precision has been always a huge problem for this type of measurement. So to do the assay precision, we follow a procedure of the CSI, is in particular the EPA 5A2. And how this works is what we have to do according to the procedure. We have to we need to have 2 lots of kit control and serum samples tested twice per day in duplicate for 20 days using 2 different analyzer, XL analyzer instrument. And as you can see, with kit control 1 and 2 at 2 different levels, as well as with the 6 sample, we obtain a single-digit CVs percentage across the measuring range, which is quite remarkable because all the other assays you will see are at least double digits, particularly in the low end and the high end of the measuring range. Then the second important characteristic of the assay is precision, for sure, and combined to the assay sensitivity, that in the low end are really dictating the precision as well. So sensitivity and precision are combined. Here, just to give you an example, we have the LIAISON XL, the precision of the XL in across the range, particularly in with more point in the lower hand, in the lower part. And then we have different methods, method A, method B, method C. And as you can see, the CVs of all the other methods in below 30 picogram per ml are extremely high, up to more than 20% in one case, and then in between 10 to 15 with all the other methods. Whilst even in at concentration very low, at 5 picogram per ml, we had CVs below 5%. So next one is sample equivalence. This is was meant to make sure that if you're taking plasma, EDTA, or SST in serum, or lithium versus serum, basically the matrix of the sample does not make any difference. And here are the data reporting the correlation are extremely high. So EDTA plasma versus serum was 997. SST versus serum is 991. And lithium heparin was 998 versus serum. Typically on 50 sample to do the sample equivalence. Then the other data that we had to produce to be included in our instruction for use was the construction of a reference range. The reference range was established per the CSI guideline C28A, and to do that we collected 123 serum samples from adult subjects of different seasons, so winter and summer, and different geographical location, different latitude in US, so Houston and Minnesota. And then in terms of inclusion criteria, we selected patients with normal values for PTH, calcium, and TSH. And by using this methodology, we had a normal range with a mean value of about 50 picogram per ml, and with an interval of the 95%. So the lower limit of the reference range was 19.9, and the upper limit was 79.3. So here is remarkable that in between the lower limit of the reference range, which is about 20, up to the limit of quantitation, which is 5, we have a certain range, and we have practically 15 picogram in terms of range, and in that part of the range we are extremely precise. And you can imagine for all the diseases in which you have to look to the low vitamin D, this is extremely important for understanding what happened from the clinical standpoint to a subject, to a patient. The next step, the next analytical characteristic is about the method comparison. In the our extraction for use in US, we had to compare to an approved kit, and one of those is the radium assay that was used as a predicate, and using the Deming, we have a correlation that was within spec, so it was 0.918, and that was done on the 140 sample with both spike, with a portion of the sample were spike and diluted within 10%, and all the rest were controlled. But then, since we understand that the mass spec is considered an high-order method with respect to accuracy, we also evaluated the assay against the mass spec. And also another reason that was inducing this effort was that right now there is a global 25-hydroxyvitamin D standardization program that is based on the mass spec as well. So we decided to go ahead and do all the network comparison also against the mass spec. And to do that, we selected two mass spec, two mass spec based on two independent methods. One was done at the University of Washington, in which, from what we understood, they are using the isotope dilution method based on a immune affinity enrichment before the mass spec with a monoclonal anti-125. And then there is a step of 125 derivatization. And then we were using a second method that is done by a commercial lab in US, Esoterix. We don't know exactly which is the method, but we know that it's different from the University of Washington, and it's more based on HPLC separation. First of all, we compared the two mass spec. So the mass spec of Esoterix versus University of Washington, and we found that the two methods on 157 serum sample, including normals, diluted sample, and also pregnancy samples, had a very good correlation. So the R was 97, the slope was 94, and the intercept was minus 368. And also with the passing bubble, the regression analysis demonstrated good agreement between the two methods. So the slope was 94 and the intercept was 353. So then once we established the two methods are kind of equivalent, we compare our product, our assay, to the University of Washington. To do that, we selected 174 serum samples, again including normals, diluted, and pregnancy. And also in this case, using the linear regression, we had a very good correlation. So the R was 0.98, the slope was 1, and the intercept was minus 459. And the passing bubble also show a good agreement between the methods. The slope was 99.99, the intercept minus 126, with the mean bias of minus 2.4%, with the limit of agreement in between minus 30.7 to 25.8. Then we did the same, another comparison then of the XL to the other mass spec, so the Esoterix. In this case, we were using 157 serum samples. Using the linear regression, the correlation was also very good. 0.95, the slope was 0.99, and the intercept 8.44, and the passive bubble was also showing good agreement. So 107, the slope, and the intercept minus 0.48. So that means that the assay is very accurate and reproducible and very similar to the mass spec. And here, the next slide report the DEQAS data that were collected up to the January 2016. Here you may, at this point in January this year, we had 70 users, 70 participants reporting the data. And here, to compare our product, we are putting, just indicating the histogram, because this is what we are allowed to present with our product, the RIA. In the meantime, the number of participants, of course, has been reduced with our product, and the mass spec. Here, the point is that the 125, as you can see here, is the most precise assay across the board. And because here you see the level of imprecision around the expected point. And you will see that bar of the imprecision is lower with our assay to compare to any other assay that is used. And also the 125 is quite similar to the mass spec. In fact, next slide is showing which is the trend. Actually, we include in this slide also the data that were collected up to July 2016. Since from January 2013 to July 2016, against the mass spec, we have a slope which is 1.06 and the R which is 0.94 with the mean bias of 0.2%. That is indicating that our assay is equivalent to the mass spec and is more precise. Finally, as soon as we launch the product is being largely used in Germany, in particular by this group of the University of Aachen. And then soon after the launch of the product, they publish on Clinical Chemical Laboratory Medicine, CCLM, end of the 2004, the first paper in which they were reporting the experience of using this product to quantify the 125 vitamin D. And there are, in the paper are presented, again, analytical data, but also including this interesting data in which basically what they did they select patient with biliary atresia, chronic renal failure, high-level athletes, were soccer player, normal clinical sample, pregnant woman, primary hyperpara, and sarcoidosis, because basically with this patient, you will be able to see whether the biliary you would expect to be low, as well as the chronic renal failure in vitamin D, as well as the high-level athletes. Then you have normals that, as I saw before, they are ballpark from 20 picogram per ml up to 80 picogram per ml. And then you have the high vitamin D, 125, so pregnant woman, primary hyperpara, sarcoidosis. And these 7 group of patients are placed in the right buckets in terms of concentration of the 125, and in a very precise way. So here in C1 there is our product in the C lane, and the L1 is the the mass spec. This is method AB-SHEX of waters. And as you can see, for instance, in group 1 of the biliary atresia, the value of the 125 with XL was extremely low with a very high precision. Whilst with the mass spec, you had a dispersion around the mean. And so you may classify patient in a different way. So you cannot classify them as for having a very low 125 vitamin D. Then going to the second group, the chronic renal failure, you see again in the C2 with the LIAISON, a very precise group of patients mapping in the low range as compared to the mass spec where you have results pretty much across the board. And then we have the high-level athletes, again, very low, very compact distribution as opposed to the mass spec, and so on. So what we did when we saw also data, this data, we decided to start proof of principle study and to evaluate then the clinical utility of the 125 measurement, particularly in the when we are in situation of low 125. So in conclusion, I think that today I hope to convince you that our 125 assay has really an unprecedented analytical performance, and we think this is because it does not require any pre-analytical step as opposed to the mass spec or the RIA. It is based on a competitive format rather than the competitive format, and the advantage of the competitive format is that, for instance, the competitive format are usually, particularly in the immunoassay, are kind of flat at both ends of the measuring range. So when you have low 125 or high 25, that causing problem of the precision. And these results are also showing the very high precision and accuracy on the low end, well below the lower limit of the reference range that was 20 picogram per ml. Also, we have the opportunity here with this assay to have an accurate classification of patient, and also which is far better than the precision that is achievable by mass spec, which is quite imprecise, particularly in the low end of the measuring range. And also this latter observation has been confirmed in independent study published by the group of Aachen. So I think for today I'm finished. I'm ready to receive questions from you. Thank you.
Thank you, Dr. Bonelli, for your informative presentation. I would also like to thank our sponsor, DESORIN, for making today's educational webcast possible. One final reminder before we close, you can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Bonelli will answer all your questions via email. Today's webcast will be available for on-demand viewing through February 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you for joining us. We hope to see you next time. Goodbye.